we were looking at chemolithotrophy ideas. And so this is an example where we're looking at inorganic electron donors. So hydrogen gas is a popular one with microbes in many, many habitats because that's a popular gas that's produced as a waste product in fermentation. So if there's a nearby microbe saying, hey, I can use that. So they're gonna strip off electrons and get it into this electron transport chain process. And if they're really smart microbes, they'll try to snag oxygen as the terminal electron acceptor. So the point is here, I'm not asking you to memorize step by step. It's just a visual trying to get across the point that we do have an electron transport chain. It's just that we have an inorganic as the donor of the electrons. And it can be oxygen, but it can be something else as well. But the game is to create a situation where they have a little bit of free energy to pump protons. So they always link it to this little bit of energy, so we'll pump a proton, and okay, a little bit here, and I'll pump another proton, and then they get this proton gradient. And the reason that they do this, I mean, haven't you ever wondered why is it such a complicated thing? You know, why don't they just go from hydrogen to oxygen, call it good, not have all these intermediate steps? Basically, it would cause the cell to burst into flames. It's too much energy released all at once as heat. And so they parcel it out in these little snippets of free energy so that they can control the heat generation and allow for a controlled reaction to get you this proton gradient. All right, so we talked about that. And then we looked at some options with nitrification. Sometimes they'll use nitrate. Often did you do that in lab last Thursday? Nitrate reduction, does that sound familiar? Maybe you'll do it this week. I know, since I'm sick, I forget which is which. So anyways, we talked about that last Wednesday. We looked at methanogenesis, where all of the rumen contents, where they're doing that. So I think we made it through all of that. So this is new material now, I believe. So this topic they refer to as light-mediated ATP synthesis. And that may sound like photosynthesis, but technically it's not. So this is not photosynthesis, but it's a really cool strategy. Halobacterium, Salinarium has this ability. This is a fun microbe to work with in the lab because they exist. Well, it's kind of giving it away. Salinarium. Where do you think they might like to live? Salt, really salt. So if you've ever flown, which I have not done, but I've seen aerial photos over California, where you see those salt flats where they're evaporating for salt, and they have those beautiful colors. It's a lot to do with these types of microbes. They're in that salt flat and they're growing on it because they love salt. So when I used to teach BI 100, I had to try to get the students to experience what DNA is in lab. And so I purchased this microbe because it loves such salty conditions. All we had to do to release the DNA was put it into pure water, and the cells would burst and you get all this precipitated DNA out where the students could actually visualize the DNA. So you didn't have to use any nasty chemicals. Like if you've ever extracted DNA before, sometimes you have to use really nasty chemicals, but I didn't want BI 100 students that are non-biology majors touching that kind of stuff. So this was a very easy one to work with. But anyways, the reason it's so well adapted to salty conditions is it has this, it has this ability to use light energy. And it's called a bacterial rhodopsin protein, often just brought down to BR. It's located in their cell membrane, and then there's a retinal bound to it. So this may sound kind of familiar, rhodopsin, retinal, because our eyes can function in a kind of a similar way in some regard to some of the systems that we have for capturing light. But anyways, it's a carotenoid-like pigment, And what it does is it absorbs light energy. And as it's doing this, it creates a situation where you can get a proton gradient or sodium gradient. And of course, that's linked to ATP synthesis. I'm going to show you a picture. This is in words, what's happening, but let's look at a picture. I think the picture makes a little bit more sense. So here's the cell membrane, and you have the bacteria rhodopsin, and the purple is the retinal, carotenoid-like pigment. And so anytime you see this squiggle line arrow, and then HV 570, that's saying sunlight energy. And in particular, it's around 570 nanometers of light quality. And what it does is when the retinal absorbs that, it changes the shape from trans to cis. Remember from organic chemistry, every time I hear trans to cis, I start having tremors and nightmares. But anyways, trans to cis conformation, and that's enough to get a little bit of proton pumping across the membrane. And so that's what they're trying to show here. Protons are getting pumped across as that transformation happens, that physical shift after it absorbs light energy. So if it keeps doing that all day long, you get a nice proton gradient. And then here we're showing the influx of the protons coming back through the ATP synthase.
It's changing the shape of the retinal pigment.
Yeah, so photoreceptors like retinal, they often change their shape in order to get into the signal transduction pathway, I guess is the way to think about it. In this case, they're linking it though to this idea of photon pumping. In the shape shifting mechanism, it allows for proton pumping. Okay, so technically this is not photosynthesis. If you've taken any class, teach photosynthesis in Plant Kingdom, Plant Phys, this is not it. But it is a way for a pigment, a photoreceptor to absorb light energy and get something to happen. In this case, it's a way to link it to ATP synthesis. All right, so this is the line you draw. This is the end of the exam material for the exam coming up on Friday. So I do still want to talk just a little bit more on metabolism, and I am now going to shift to photosynthesis. This topic is a little beefier, so I don't want it on the Friday exam because you only have till Friday. So let's put this on to the next exam, exam 3. So now I am not going to go into huge detail on photosynthesis per se, like I do in Plant Phys. However, there's some key ideas I want you to grasp. And then one of the ways I think to approach studying this material is to do a compare and contrast. I'm going to show you that microbes do it different. They don't all do it like plants do. And some do it very differently than others. And yet it all falls under the category of photosynthesis. So they were the ones that came up with this first. It's not like plants invented photosynthesis. That just didn't happen. You have the microbes starting off with this, a couple billion years ago, and through refinement, natural selection, adaptation, you start to see a more sophisticated version showing up in the plants. So this is a big picture summary of photosynthesis. And a couple of things. So this is looking at, typically they show this in a plant class, but what is this green thing? What organelle? In chloroplast. But remember, the chloroplast was once a free living cyanobacteria, an ancient cyanobacteria. Okay, so cyanobacteria that we currently have on this planet do something very similar to what's being shown here, if not identical. So you have the idea of chlorophyll, pigments, absorbing light energy. So that's what they're showing here. All right, so you have that going on. Why is that important? Well, that allows water, electrons to get stripped off of water. And so it's a very amazing process where you have the ability to strip electrons off water. Water does not like to give up electrons. It really doesn't like doing that. And so you have to hit it with a lot of energy in order to extract the electrons. And then that kicks in an electron transport chain process, right? Now, they're not showing the details of that, but what they're showing is that you have it happening in this internal membrane here. This is a membrane inside the chloroplast, the thylakoid membrane. And they're showing ADP and NADP plus coming in. So in this process right here, you do have an electron transport chain. I'll show you in a different picture. And at the end of the electron transport chain is NADP plus, waiting to get an electron. And then once it does, it gets reduced to NADPH. So by the time the electron transport system has run its course, you get NADPH, which is great. That's really good for the microbe. That's a reducing molecule. Now it can participate in second part of photosynthesis, which is the Calvin cycle. The other key thing, it's an electron transport chain process that's cooking along in that thylakoid membrane. So that's linked to proton pumping again. All right, so just like what we've been talking about, if you can figure out a game where you can get an electron transport chain going from donor to receptor and do that in the correct way where you get some free energy, bits of free energy, then you can pump protons. And that's what we see again in photosynthesis. So then that allows you to get your lovely ATP. Your proton gradient gets established and you generate ATP through that. So two huge things coming out of this. You get NADPH, a reducing molecule, that's really good, and some ATP. And then they're trying to show that those two products of the light reactions go into the Calvin cycle. And they're necessary because at key steps in the Calvin cycle, you do have to expend ATP. You do have to donate some electrons, and that's what NADPH does. That's its job, is to, here, I'll give you some electrons in order to keep that Calvin cycle going. And so the Calvin cycle is where you actually see the carbon dioxide getting brought in. And so if you're an autotroph, you can take in CO2 and make your sugars. And so cyanobacteria do this, plants do this. And the Calvin cycle creates a situation where they can, it actually feeds into a different pathway. That's why they're showing this swooping line out of the chloroplast, because what happens is one of the chemicals, intermediates in the Calvin cycle can get shifted out into the cytoplasm of a plant and make sugars. And so this is sucrose that they're actually making. When you think about how plants are doing it, they make sucrose. Okay, so this is the big picture idea where they're talking about what's going on with the light reactions, what's the goal, and then that feeds into the Calvin cycle, and the goal is to make sugars. Okay, so when you look at microbes, it depends on the microbe and what habitat have they adapted to, what version of photosynthesis. So the one I just talked about, plants do that, cyanobacteria do that, but there's other photosynthetic microbes that don't do it like that. And so those are the ones you never really hear talked about in other classes when they mention photosynthesis. So there's different versions out there, but you can say, well, they all seem to have some kind of antenna system. And if you use the antenna system on your TV, that's why they named it an antenna system, because you're trying to capture those, the TV waves that are coming through and get it into your TV. Same thing here. What they're trying to do is capture sunlight energy, little different wavelengths of sunlight energy, and get that brought in. And so there's an antenna system. It's just made of different pigments that will capture sunlight energy. And then all that energy has to get to what we call the reaction center, right? The reaction center, which is a complex. It's complicated. We're not going to get into it. But what happens is that this reaction center complex, microbes can have one reaction center complex or two. And basically, they create a situation where they can get an electron transport chain hooking along. So some version of an electron transport chain or electron transport system. And then they'll have energy carriers that will get involved in this too. And you end up, you know, a big energy carrier at the very end when you get NADPH. So there are some commonalities, but there's some definite differences too. So this is just a visual trying to get you to think about the antenna complex. Basically, it's just a lot of pigments that capture sunlight energy, a little different wavelengths, so they are pretty efficient. And then we tend to say the energy funnels down to the reaction center. And it's really the reaction center where you see things really start to happen. So here is a little bit more detail of what's going on inside a chloroplast during photosynthesis. Now, again, I'm not going to ask you to memorize step by step in this class what's going on. However, again, a couple of key things. So if you think about cyanobacteria and plants, this is their version of photosynthesis. So what's important here? Two photosystems. So you need to know cyanobacteria and plants they have two photosystems. And what they're trying to show here is that the black arrows are the electron flow going. Okay, so you see that there's more than just those two photosystems in the electron transport chain. You got some other players in the game here doing that hot potato, you know, donating off the electrons to the acceptor on down the line. Ultimately, when cyanobacteria and plants do their electron transport chain, the ultimate donor, we say, is water. So water is getting stripped of electrons. And then what's really going on here, do I have it? So in this photosystem too, that's one of the reaction centers. There's a reaction center in there. There's a reaction center in photosystem 2. And then there's a different reaction center in photosystem 1. And in that reaction center is a very special chlorophyll pigment. What happens is that chlorophyll pigment will get that sunlight energy and it excites an electron of the chlorophyll. And so the chlorophyll pigment gets one of its electrons excited, and then it gets donated off to an acceptor. And that kicks off the electron transport chain. But now that chlorophyll is like, I'm missing an electron. I just donated one off. I need another one to feel chemically happy. And so what happens is it'll turn to water and say, give me one. All right. And then water gets extracted, the electrons get extracted from water, given the chlorophyll, and then chlorophyll's like, okay, I'm back to my normal electrons. Then it gets hit with sunlight energy again, and the whole thing happens all over again. And it's picosecond flashes of reaction pathways. It happens so fast. It's amazing, amazing process. And so we have this chlorophyll, the special chlorophyll in the reaction centers with their electrons getting excited. So in words, so we have two photosystems. We abbreviate them down to photosystem 1 and photosystem 2. And they have those special reaction centers in them. And then that chlorophyll, where's the chlorophyll? Where is that slide? Here, let's go to this one. Chlorophyll in the reaction centers absorb that light energy, and that excites one of their electrons, and it gets donated off to an acceptor. So now that chlorophyll is like, I'm missing an electron. I don't like this. I need an electron in order to be chemically stable and happy. And so what happens is that water in where the photosystem 2 is sitting up there, water will be used as the source of electrons. So the photosystem 2 can extract electrons from water that gets given back to chlorophyll, and then it gets hit with light energy again, and the whole thing happens. So you can get electrons zinging down this electron transport chain process in really quick order. So water, we say, is the ultimate electron donor when you look at cyanobacteria. And it's the chlorophyll pigment that's its electron is getting excited as well in order to start the flow of the electron transport chain process. And then through that electron transport chain process, you get a proton gradient. You get there's T steps in the process where a little bit of free energy is there to pump the protons across. And so you get this nice proton gradient.
Sorry, what's the thing that gets excited? Oh,
So there's a chlorophyll in the reaction center, a chlorophyll pigment in the
Chlorophyll
reaction
pigment.
center. Yeah, so there's a chlorophyll pigment. And so its electron gets excited and gets donated off. And then it has to grab an electron from water to fill the missing spot. And then that just keeps happening over and over and over in really quick time frames. And so then you get that proton gradient, which gets you eventually to the ATP synthesis. So it's very similar to what we've been talking about, where you link an electron transport chain process to a proton gradient, which gets you your ATP. It's just that they have this very special ability to use light energy to get this all cooking along. And so cyanobacteria are doing this, plants are doing this. And the ultimate goal really is to get a proton gradient, to get your ATP, and then to get NADPH formed at the very end of the electron transport. The other thing to think about is that if you take electrons off of water, what are the other products? So you've got electrons flowing into the electron transport chain, but it leaves you with protons, which is good. And then oxygen, which is good for us, because that allowed us to evolve and breathe. Okay, but from the cyanobacteria, they're not too concerned about the oxygen. They're like, no, if you're gonna strip an electron off me, at least I can get some protons building up. And so the protons coming off of the splitting of water actually helps with the proton gradient. It helps build that proton gradient over time. So going back to here, they're trying to show that where if you look at what's going on with water, they're showing that you do get protons. And then the proton pumping at key steps. And then I wish they would have drawn a little bit, just, more protons building up over time. So you get a nice proton gradient, and then you see the protons going through. And so the Calvin cycle is right here, right where ATP gets synthesized, NADPH gets synthesized, and that feeds immediately into part 2 of photosynthesis, which is you got to get your sugars. You got to be able to take in the CO2 and get your sugars. Questions? Let's look from your book. I think they give a image something like this, or how cyanobacteria and chloroplasts inside plants run it. And so again, you're seeing that there's 2 photosystems. Okay, 2 photosystems. And then you got some other players in there. And here you follow the blue arrow for the electron transport process. And then you see your water. So we call this electron transport process the linear flow. Okay, so far that's all I've been showing you all. When we looked at aerobic respiration, anaerobic respiration, all of that, it's this linear flow of electrons from donor to acceptor until you get to a terminal acceptor, which in this case is NADP plus. That's your terminal acceptor. So we call this a linear flow. And I'm pointing that out because some microbes don't do a linear flow. They do a circular flow, which is really weird. So again, you don't have to know all the players, but I do want you to know that there's two photosystems involved. You have an electron transport chain system where the electrons are flowing. Water is the ultimate donor. NADP plus is your ultimate acceptor. And then you got that proton gradient getting established through it, through this whole flow. This figure up top that you see, if you tilt your head sideways, they call this the Z scheme, because if you tilt your head sideways, it's kind of a Z flow. Don't get lost in that, but they do talk about it in the book. They call it the Z scheme. It's just trying to show what they've done here is they put it on the reducing potential tower here. Where, remember, if you have something up in the negative, like that redox tower we looked at, if you get this electron, up into the negative portion, that means it's really happy to get donated off. And so once you hit chlorophyll with light energy, it goes up into the negative portion, so to speak, in the sense that it's nicely excited and can give off an electron. And then you can see that the energy starts to fall down here on the scale. But then in photosystem 1, you have another chlorophyll there in that reaction center, also absorbing light energy. And so then it gets excited again and then gets donated off. And so they're just trying to get those chlorophyll in those reaction centers, get it up into, get it excited enough where it's in a negative reducing potential frame of mind where it's ready to give off that electron. Okay. Okay, so chlorophyll. If you've never looked at chlorophyll, this is a drawing of chlorophyll. And so it's this is the so-called porphyrin head region of chlorophyll that kind of sticks in the membrane. And then this is the glycol tail, they call it, that will kind of stick off the membrane. And there's different versions of chlorophyll. Like the ones we usually talk about the most is chlorophyll A, but there's chlorophyll B, there's chlorophyll C, there's chlorophyll D. It just depends on the microbe or the plant, what they're going to have, where they're living. And magnesium is sitting right smack in the middle. So magnesium is necessary in order for chlorophyll to function. If the plant or the cyanobacteria doesn't have enough magnesium, can't do it. All right. So chlorophyll A is the one that we usually focus on when we talk about photosynthesis, because we tend to teach what happens in plants in most classes. So chlorophyll A is found in plants and algae and cyanobacteria, and then, like I said, there's chlorophyll B and C and D. Those are found in a variety of different plants. And then there's bacterial chlorophylls. So bacterial chlorophylls, these are different versions of chlorophyll that show up in different groups of bacteria that do photosynthesis. And so they absorb slightly different wavelengths of light, which is nice because they're not always competing for the exact same wavelengths. Then you have all these accessory pigments that are helpful in the antenna systems. They capture their own particular wavelengths of light. They get that energy down, so to speak,
into the reaction center where those very special chlorophyll pigments are there. And for many organisms that do photosynthesis, just like, you may have been told by your parents, too much of anything can be bad for you. It's the same with organisms that do photosynthesis. If they get too much light energy hitting them all at once, it can cause issues. It can damage their DNA and create free radicals and just be a problem. And so these accessory pigments also can help deal with that. If there's too much light energy, they can capture some of that and kind of convert it to heat, which is better than having their DNA get broken down or their proteins getting broken down. As far as I know, I've not seen an exception to this. Any photosynthetic organism, they'll have carotenoids somewhere in their system of doing photosynthesis. And those carotenoids, again, they're very helpful to also protect. They're involved in capturing light energy for photosynthesis, but they also help the organism not get too much sunlight too fast all at once. It will convert it to heat if necessary. And then, phycobilins are another group of pigments that can show up in different cyanobacteria. So different pigments to absorb different qualities of sunlight. It varies depending on where the microbe lives. So if you have cyanobacteria that prefer a deeper depths in the ocean, for example, they'll have the ability probably to absorb more reddish qualities of light than anything else. And so it just depends where they live, what kinds of pigments they end up evolving. But if they can absorb slightly different wavelengths, so these are spectral outputs of some of the pigments. And so where you see peaks, that means that's where those wavelengths of light is what they've most adapted to in terms of, I really like that quality of light. I'm really good at absorbing it and using it for photosynthesis. So you can see phycocyanin is a pigment that's different than chlorophyll A. And then the bacteria chlorophyll A, you can see, are different compared to all the others. I mean, so it's nice to not have them all competing for the exact same quality of light. It allows for photosynthetic organisms like bacteria to hang out really close to each other, and that's okay. They don't have to fight, so to speak, for the same quality of light. They can absorb slightly different, and they can all be happy living together. So here again is just trying to show you that you don't have a ton of overlap on the peaks, which is good, because then you can have consortiums, mixtures of bacteria, like those stromatolites I talked about, where you have these big mats of bacteria where they're actually piled on each other, and yet they're still being capable of doing photosynthesis because they are able to use different qualities of sunlight. So it works really well for bacteria. Okay, so bacteria can do photosynthesis a little differently from plants. So if you've learned photosynthesis in like a plant phys class, that's what's going on the blue side here, where plants take in CO2 and then they run, they're autotrophic and they can make sugars from that. Lots of bacteria, cyanobacteria do that, sure. But then there's some funky bacteria that can't do that. They can't take in CO2. So they're weird. They have to use some kind of organic carbon that they'll bring in for their carbon, just like we do. We have to take in some kind of organic carbon. Unfortunately, we can't take in CO2 and make sugars. So I'll mention a little bit about that. So here is another interesting comparison. So here, they're breaking it down into anoxygenic or oxygenic. So if you've taken a plant phys class, you're used to talking about it like what the green box is showing. So, and it's what we just talked about. Water is the ultimate electron donor. The electrons go into an electron transport chain. It allows for production of ATP. And cyanobacterium plants can take in CO2 and make their sugars. Great. Why do they call it oxygenic? Because in the process of stripping electrons off water, oxygen's made. oxygenic. Genic is to create, Genesis, so creating oxygen. But again, from a plant's perspective, cyanobacteria, that's not really their goal. They don't really care about oxygen. We do, but they don't. They're going for ATP and NADPH. That's what they care about. Then there's some funky microbes that do very different. They do anoxygenic. So without the creation of oxygen. So if you follow that thought out, okay, if they're not creating oxygen, then they must not be using water as their electron donor, their source of electrons. They can't. Because anytime you take electrons off water, you're going to get oxygen. So if they're anoxygenic microbes, that means they're using something else. They're not using water, and that's what you can see here in this particular schematic. They're showing hydrogen sulfide is being used instead of water. Okay, so anytime you hear anoxygenic photosynthesis, you know that means water is not the source of electrons. Can't be. So here, if they strip electrons off of hydrogen sulfide, you see you're left with a sulfate molecule of some sort. So that's definitely not oxygen. But it's the same basic idea. You strip electrons, in this case, off of hydrogen sulfide, you get your electron transport chain going. That allows for some ATP. They're not showing the NADPH, but that's there too. And then you can take in CO2 and make your sugars. So anytime you see CH2O written that way with the parentheses and N, that's the basic building block of a carbohydrate. So there's some microbes that do photosynthesis, but they don't do it creating any oxygen. And in fact, they live in areas where there's no oxygen primarily. So here, you know, when you look at a chloroplast, there's all that internal thylakoid membrane. It's just jam-packed full of membrane material. Microbes can do their version of that. So cyanobacteria certainly have their version of an internal membrane, similar to what you see in a chloroplast in a plant. But then there's these so-called purple bacteria that do photosynthesis, and they have internal membrane systems too. And depending on the species, it can look more stacked up like that, or a bunch of vesicles. And so this is where I kind of smile, and I mean, this is kind of like organelles. And even though you're taught in basic biology that prokaryotic organisms don't have organelles, and yet it kind of looks like an organelle. But they won't say that yet. All right. So, okay, what happens if there's just one photosynthesis, not two? So let's go back a billion or so years before cyanobacteria showed up, and think about a little bit simpler forms of photosynthesis, where they didn't have two photosystems yet, they just had one. Now there's still debate what came first, photosystem 1 or photosystem 2 in the process of evolution. It's still a debate. Last I read, they're leaning toward photosystem 2, but it's still a debate. Anyways, there are some microbes like green sulfur bacteria. That's their actual group name. Green sulfur or chlorobia. You don't need to memorize that per se. Know though that there are some bacteria that just do photosystem 1 and some other players for an electron transport chain. They really love hydrogen sulfide gas. If they have that, they'll use it. But if they don't have that, they can find something else typically. Often it's an iron source. So again, these are organisms that are adapted to areas with high hydrogen sulfide, and that can be pretty common actually. There's a lot of places on Earth where there's a fair dose of hydrogen sulfide. But basically it's the same idea. They have this photosystem, they have this very special reaction center, and they have their bacterial chlorophylls, and they get that excited and get the electron transferred all the way down to, there's either going to be NADP plus or NAD plus sitting at the end of that electron transport chain process. And then that's good because it's a reducing chemical. It's an electron carrier, that's good. And They also, as the electron transport chain is going, they have the ability to create a proton gradient. So they link that to a proton gradient, which then gets you your ATP. All right, so photosystem 1, the green sulfur, when they looked inside the green sulfur, once again, they found very special membrane systems inside. And it's called the fluorosome structure. And so the arrows are pointing to, you can see there's many of them that are kind of pressing up against the cell membrane. So those are extra membrane material, and they're just really good at, there's pigments in there that are really good at absorbing really low quality, dim light. So you find green sulfur bacteria kind of hanging out in deeper places in the water column, for example, in either freshwater or oceans. They'll be in dimmer light. And so they're really well designed for, even though it's not the greatest bright light, they can still grab that light energy and use it to generate their electron transport chain. So this is what it looks like. This is, let me zoom in. So here is your photosystem 1. Photosystem 1. And they're showing in the reaction center, there's a bacterial chlorophyll. BCHL stands for bacterial chlorophyll. So its electron, once it gets hit with that light energy, its electron gets excited. And you can see it's going up into the negative portion of the redox diffusion potential. And you can see there's other players that will send the electron through that chain. And then here is your final NAD+, is your final electron acceptor. So you can see it's kind of a short little pathway. Let me show you this version too. So this is the same thing. This is your photosystem 1. And you see your hydrogen sulfide. And you can see the blue arrows are your electron flow. So simple, not so complicated as what we saw.
The one photosystem, are they only making NADPH or NADPH and not making ATP?
No, it does make it. See, again, I wish the artist and the scientist talked. Because yes, they're making NADH or ATPH, and they're making ATP, and you don't see it here. It drives me nuts. So it also is creating a proton gradient. It's just they're not showing it very well. So if you are an artistic person, you might want to be an artistic or scientific illustrator. We need people that can do that. So yeah, see, they don't show it here really either, but I have it. That's why I put it here. Bacteria using photosystem 1 also generate in that proton gradient. It's just the artist didn't show it very well. Okay, one more idea. Where is it? Okay, so that's photosystem 1. And what do we see? We see a linear flow of electrons. The ultimate donor is, in this case, hydrogen sulfide. And then it goes linear all the way through to NADP plus or NAD plus. It does generate a proton gradient. So you get both ATP and either NADPH or NADH. Okay. Let me just quickly introduce this other idea to compare it. Really quickly. Some microbes only have photosystem 2. They don't have photosystem 1, just photosystem 2. Now, if you look at the blue electron flow, the blue arrows, this is one that's circular. So this is really fun. We have a system where they get an electron excited in the reaction center, and it just keeps zigging around and around and around and does this weird, funky circular flow. But it gets them what they need. So we'll talk about that next time. So Wednesday, we'll finish up this idea. I'll talk a little bit about microbial growth, which is a new slide set, and then we'll have time for questions about the exam as well.